Okay, we're back and we're going to cover now the last two of our four major signal transduction pathways. We'll begin with another phospholipid pathway. So, another pathway that involves lipids of the inner leaflet of the plasma membrane. And we're going to begin again with PIP2. And here's our close up view of that inner leaflet phospholipid. And if you recall, we noticed in our last signaling pathway that PIP2 could be acted on by a lipase enzyme that would basically clip off or cleave off the hydrophobic tail and then liberate the polar head group. Well, it turns out that phospholipids like phosphatidylinositol bisphosphate, PIP2, can also be, oddly enough, phosphorylated. And you may be thinking, well, that seems strange because we've always thought of phosphorylation, happening when an enzyme transfers phosphate group to a protein substrate. We haven't yet talked about this happening to a lipid, but hey, turns out lipids can be phosphorylated too, at least their polar head group can. So the enzymes that perform phosphorylation of a lipid are also kinase enzymes, but instead of being protein kinases, they're phosphatidylinositol kinases. So we usually abbreviate it as PI3K for phosphatidylinositol 3 kinase. So this is the enzyme that can perform this operation, adding this phosphate group to the PIP2 substrate. Another way of drawing that, as the cartoon kind of indicates, is we could draw it like this. So PIP2 is the substrate, PI3 kinase is the enzyme, and it produces PIP3. And you can see PIP3 is identical to PIP2 except for that extra phosphate group that's now there on the polar head group. And remember, this is facing the cytosol. Okay? So one of the really cool things about this PIP3, if you will, pathway is that it's very critical for cell survival and prevention of cell death. And next week we're going to hear a lot more about this pathway and see how it regulates a process called apoptosis. All right. Here's a look at what happens downstream of PI3 kinase activity. In other words, once we have PIP3 present, we can follow the signaling pathway downstream. And what the artist has done here is they've drawn for us, two PIP3s produced by PI3 kinase phosphorylation of PIP2. And remember, in reality, the artist has drawn it in a very simplified manner so that we can just sort of follow the logic. But in reality, there are going to be hundreds to thousands of PIP3s converted in a small local area of membrane. So we're just looking at a little snapshot, a simplified version, so we can get the logic of the pathway. So one of the features of this pathway is docking of cytosolic proteins to the membrane. This is very much analogous to what we saw in the receptor enzyme situation. And there you remember it's phosphorylated tyrosine that's acting as a docking site for proteins that contain an SH2 domain. Well, PIP3 is very similarly a docking site for proteins that contain a particular domain, but now instead of the SH2 domain, it's what we call the pH domain. And this has nothing to do with acids and bases. It's an acronym for plextrin homology domain. Okay, so pH domain is the way we refer to this part of the protein that is able to physically engage PIP3. So the way this would work is this protein, PDK, and this protein, AKT, are normally down here soluble, freely moving around the cytosol. Well, once PI3 kinase has produced these PIP3 molecules, these proteins are now brought up to the membrane. They're going to literally diffuse and bump into that PIP3 and then be able to latch onto it. So they latch on. And what we've done here is we've brought substrate in proximity to its kinase. PDK is a protein kinase, so it phosphorylates the AKT protein. And you can see these two phosphate groups that have been transferred from ATP to the AKT substrate. Now, once this happens, once AKT is phosphorylated, it changes shape. It lets go of PIP3, and that's what the arrow is indicating. And it now moves as a soluble activated enzyme into the cytosol to then modify substrates. And we're not going to follow it any further downstream now, but I'll preview for you one of the topics of next week's lectures. And one of the things we know about AKT is that one of its substrates, arguably its most important substrate, is a protein called BAD. And you have to love this, a protein named BAD. And its name is fitting because BAD is bad for cells. It leads to cell death unless it's phosphorylated by AKT. And then we prevent cell death. So survival factors, growth factors, those survival ligands that I alluded to in one of the earlier slides of part 1, what those survival ligands do is they set off a signaling cascade that activates PI3 kinase, activates PDK, activates AKT. AKT phosphorylates BAD, and that prevents cell death. If BAD is instead in the unphosphorylated state, then we allow for cell death. More on that next week. We're just previewing it now. Okay, so that's the end of the third major pathway. If you're keeping a running tally, pathway 1 was that cyclic AMP PKA signaling pathway we talked about. 2 was the first of 2 phospholipid pathways. I'd call it the PIP2 pathway, where phospholipase C cleaves PIP2. And then the third one that we've just talked about is what we would consider the PIP3 pathway, where PIP2 is phosphorylated instead of cleaved and turned into PIP3. So let's move on to the last of our 4. Okay, the last of the 4 major pathways is this one. It's known as the ERK-MAP kinase pathway. And it's really strange nomenclature. There are really 2 ways of referring to the pathway. Some people will just say the ERK pathway. Some will just say the MAP kinase pathway. I've sort of hedged and given you both of them. It really depends on what literature you're reading. Sometimes you'll see it referred to this way and sometimes this way. It's worth knowing that these are synonymous and that there are 2 different ways of referring to the same pathway. Somewhat complicating things, there are also 2 ways of referring to each of the proteins in the pathway, but I won't bother you with that layer of complexity. I'll kind of keep it simple for you and just choose one bit of nomenclature. And mostly I'll do what the artists have chosen to do on these slides. So let's introduce the pathway. This is one of the most important mitogenic pathways. A mitogenic pathway is one that increases cell division. One of the reasons this pathway is so important is because of its role in some cancers. You probably know that cancer is essentially uncontrolled cell division. So any dysregulation in this pathway, this mitogenic pathway, can lead to cancer. Okay, and we'll see a couple of examples of that, particularly in colon cancer, as we move through this material. So let's introduce the pathway. And there are lots of ways to get this pathway started. It's almost like a mix and match modular type of situation. But I like what the artist has done here because this is probably the most common way to tap into it. And that's via a peptide ligand. They're labeling it here a growth factor, but one of those peptide ligands we've talked about before, binding to a receptor enzyme. typically a receptor tyrosine kinase. And the artist has eliminated some of the upstream stuff, which I think is fine for now. But suffice it to say, we get dimerization, cross-phosphorylation, and activation of the pathway. And then these arrows are really representing some of the early proteins that end up docking on this activated receptor enzyme. We'll flesh out that detail later. Let me just give you the broad strokes now. So a couple of steps here in between the receptor and then this protein here called RAS. RAS is a very important oncogene. An oncogene is a gene that has been linked to cancer. And RAS is a GTPase. A GTPase is an enzyme that can hydrolyze GTP. Well, the other thing that RAS can do, in addition to its ability to hydrolyze bound GTP, is it can activate the ERK-MAP kinase pathway. So it's one of the early upstream components of this pathway, if you will. So RAS in the GTP-bound state can bind and activate RAF. RAF is a protein kinase that, when activated, phosphorylates MEK. MEK is also a protein kinase, and when phosphorylated and activated, it phosphorylates ERK. And when ERK is phosphorylated and activated, it phosphorylates a whole bunch of other substrates. A lot of divergence in this pathway. And we'll look at some examples of those downstream targets. Many of them regulate gene expression, and many of those gene products drive the cell cycle and cause cells to divide. So dysregulation in this pathway, where it's too active, can be very dangerous because of the uncontrolled cell division that results. And this is particularly the case for colon cancers. Many colon cancers occur because of dysregulation of RAS. And essentially what you end up with is a situation where RAS is always on, or constitutively on. Constitutive is a fancy word for constant, so constantly on. And that means that we're constantly driving all the downstream components of the pathway, including those signals for cell division. So even after the growth factor goes away and all the upstream stuff has been quenched, if we have a mutant version of RAS that's always active, it doesn't matter what's happening upstream. We're going to constantly drive what's downstream. That's one of the reasons that there's so much interest and focus on signaling pathways, because of their relationship to diseases like cancer. So speaking of RAS, let's take a look at how it's regulated. How do we increase its activity, turn it off, all that. So this is kind of neat. I think this is at least the third, possibly even the fourth time we've heard of this kind of regulatory mechanism involving GTPases. Let's take a look. So here is a representation of inactive RAS. You'll notice that RAS is anchored in the inner leaflet by a little lipid group. And the protein side of it is kind of hanging here into the cytosol, and it's bound initially to GDP. Okay. Now, the way we stimulate RAS and convert it into the active state is to do nucleotide exchange. So let's get rid of that GDP that RAS is bound to, and let's replace it with GTP. And the enzyme that does this is a GEF, a guanine exchange factor. Okay, a GEF. And if you're thinking, where have I heard about this before? Well, you heard about this with regard to the alpha subunit of the G protein. Similar kind of regulatory mechanism is in place for it. It's off in the GDP bound state, it's on in the GTP bound state. So here's another example. The other place we've seen this is with the entry of nuclear localization signal containing proteins, moving them across the nuclear pore complex, and the role of GEFs in doing that as well. It's a very common mechanism, in other words. And as long as RAS remains in that GTP-bound active state, it's going to activate RAF. and send the signal through that MAP kinase pathway, ERK-MAP kinase pathway. It'll stay active until the GAP enzyme helps to act as a cofactor. It's not an enzyme, excuse me. The GAP cofactor helps to act to stimulate the GTPase ability of RAS. So RAS is the GTPase. It's the enzyme that can hydrolyze GTP, but it doesn't do it without the GAP cofactor. Hence, we call the GAP cofactor, what that stands for, GTPase activating protein. So what it would look like is the GAP would bind to the active RAS and stimulate RAS to hydrolyze that GDP, get rid of one of those phosphates. And of course, what that does is it regenerates GDP-bound RAS. Okay, so a couple of points before I move on here. First, note that this is true nucleotide exchange. We're getting rid of or displacing GDP, replacing it with GTP. So I just want to point that out because I don't want you to have the impression that it's a phosphorylation event. Because we're adding an extra phosphate, you might think, oh, we've just had a kinase modify GDP. It doesn't work like that at all. Kinases don't modify nucleotides, okay? So instead, we have to just completely exchange the nucleotide from the di to the tri. Second, the relationship to cancer. So cancer, most forms of cancers that are linked to RAS mutations occur because of mutations in the GTPase ability, typically loss of function mutations in the GTPase ability. And so these versions of RAS that are mutated in cancer, some cancers, they don't work as well. They're not able to hydrolyze that GTP even with the GAP cofactor. And the result of this is the GTPase activity is gone or severely limited. And the result, of course, is the GTP-bound active state is maintained much, much longer than typical. And that can cause uncontrolled cell division. Here is the fleshing out of some of the mechanism upstream of RAS that we kind of glossed over earlier. And here we've really taken a much more complete look at the mechanism. So let's go through this together. So here again, this is the extracellular space. This is the cytosol. This is obviously the plasma membrane, outer leaflet, inner leaflet. Here's our extracellular growth factor ligand. It's caused dimerization of a receptor tyrosine kinase. Activation and cross-phosphorylation has occurred. Here are all the phosphotyrosine docking sites. They also, remember, serve to turn on the tyrosine kinase. So now that enzyme part of the receptor is active. So SH2 domain containing proteins are the ones that are able to interact with phosphorylated tyrosine. And there's one called GRUB2. Here's GRUB2. It latches onto phosphotyrosine of the activated receptor. And now what we're doing is creating a little scaffold. And now the next protein can join. The next protein is called SOS. SOS doesn't have an SH2 domain. It has a GRUB2 interaction domain. So now this protein-protein interaction occurs. What's interesting and cool about this is this protein interaction can't happen unless GRUB2 is anchored to this receptor. Why? Well, because it's not in the right shape otherwise. So even though these two proteins exist together in the cytosol, they're bumping into each other, but they're not latching onto each other because they're not in the perfect shape yet. GRUB2 isn't anyway. What puts GRUB2 in the right shape is the physical interaction with the phosphotyrosine that changes its shape such that now it can bind SOS. It's kind of cool. And it prevents premature association in the absence of a ligand. Very important to regulate that interaction. So what does SOS do? Well, look what SOS is. It's the GEF. It's the guanine exchange factor that's going to kick out that GDP and replace it with GTP on the RAS protein. Now we've turned on RAS, and now RAF can activate RAF, and then RAF can phosphorylate MEK, and MET can phosphorylate ERK, and then ERK can do a whole bunch of different stuff. That's the pathway. All right, let's take a look in the next slide at what ERK can do. So, as promised, here's ERK. It's been phosphorylated by MAC, and what ERK is able to do here is typically enter the nucleus. And inside the nucleus, it's able to phosphorylate a whole bunch of different things, including trans factors. So one of the trans factors that ERK is known to phosphorylate is a protein called ELK1. And here's the phosphorylation that happened via ERK. It puts ELK1 in the right shape to bind to this SRE cis element. And now phosphorylated ELK1 can attract another protein, a trans factor called SRF, and it binds to the cis element. And this duplex is sufficient to recruit RNA polymerase to drive transcription of target genes. And many of those target genes are so-called immediate early genes. These are also trans factors that regulate still other gene expression. And a lot of those gene products do things like cause the cell to go through the cell cycle faster or to never pause in the cell cycle. So I'm going to call them cell division regulators. So you can see how it's fine. It's one thing if you need the cell to divide and it's received the signal that tells it to do so. It's quite another thing for it to happen inaccurately or inappropriately. And that's what we want to try to prevent in a situation like cancer, for example. All right, so what we've seen today in lecture 17 is really the four major signaling pathways. Part 1 focused on cyclic AMP and pKa, along with PIP2. Part 2 is focused on PIP3 and the ERK MAP kinase pathway. I want you all to have the following sort of concepts, okay? First, all these signaling pathways require a ligand-receptor interaction. So that interaction is critical to getting the ability to tap into this intracellular network of proteins. Without the ligand, without the receptor, you don't get the activation of the pathway. Another thing, sort of common thing to realize, there's a lot of stuff going on here in the membrane, and then it typically activates a cytosolic cascade. And a lot of the effects in the cytosol are important. They're almost all at the post-translational level. But what is even maybe arguably more important is when one of these factors enters the nucleus to then drive transcription or gene expression, because now you get a longer-lived effect. And typically signaling pathways have both effects in the cytosol and in the nucleus. Okay? But the ones that impact the nucleus tend to be longer lived because now they're moving beyond a post-translational mechanism. Now they're turning back the information flow clock, if you will, by activating new gene expression. Okay? So now new genes that get then, transcribed into mRNAs that get translated into proteins, and the cycle continues. All right, so a lot of information came at you today in 17. One thing I always find helpful in categorizing this information is to practice drawing the pathways. So draw a receptor in the membrane, draw the ligand, and then think about the steps and think about the sequence of the proteins. What we'll do in our next lecture is we'll do a little summary of some of the stuff, and then more importantly, we'll look at experimental techniques that help us to figure out the identities of these proteins and their order in the pathway. So that's a lot of fun. We'll talk about that the next time we're together. Okay, until then, take care, be safe, and I will talk to you soon.